Hello. Good morning.
Good morning.
Hello.
Sorry that took us to
Welcome.
figure out how to use Zoom on the Surface, but I got it working.
Nice, nice. It's, yeah, hoping my webcam's been like randomly dying on me occasionally, so I'm hoping that hoping it holds up for the next hour, yeah.
Yeah, that's fair.
Switch to
So
my phone or something.
are you presenting like for a specific person, or is this more about like recording this for someone to review later?
It's for yeah, it's presented to the committee. So they have to like give approval then to submit my thesis based on this for the
Okay.
initial thesis submission. And then they review the thesis, and then there is a the defense and then the final submission. So yeah,
Okay.
it's like sort of an exam, but like yeah, it's pretty like casual and more of a formality, this one.
Okay, I see. So they'll also be joining this Zoom call, basically.
Yes, yes. Yeah, yeah.
Okay.
So these are usually done in person, but it's just been a nightmare booking any other rooms or anything lately. Hi, Tim.
Hey, Darren.
How's it going?
Good. How are you?
Good, good. This is my brother, Aaron,
Hi,
Hello.
Aaron. Nice to meet you.
up very
Nice to meet you
early.
as well.
Where are you, Aaron? Where in the world?
I'm in Vancouver.
In Vancouver. Good.
Hi, Jack.
So
Hi
early in
there.
the morning. Yeah.
A little bit, yeah, but that's okay.
Hey, Tim.
Hey, Jack.
Amazingly, that chapter appeared out of nowhere.
I'm so pleased with that. It looks so nice. The book looks terrific. Um, I had given up hope, honestly, on that one, but that came out very well.
Yeah, I looked through it a little bit. It looks really good.
Hi,
Hi,
Ed.
hey, Ed.
Hey, how's everyone doing?
Pretty good. Pretty good. Yeah, we'll just give it
Beautiful
a
image.
thank you. Yeah, thank you. I'm not actually gonna talk about the data at all, but it's a good cover image. Yeah, yeah. So yeah.
This looks
No,
like it
I'm
could win a
leaving.
competition.
I'm
You
leaving for
should,
Toronto. I'm leaving here
I
around.
I did I yeah. I've had many submissions to the Science Expos that have made it into the
all right,
finalists, but
so
I've never
I'm
won.
doing
Until what time did that finish?
about
I
11.
want to turn off this AI thing. It just automatically
Late morning.
joins. So
Yeah, yeah. I
No.
think I
What?
just turned
is
it
that
off.
okay?
Okay.
What
I,
AI thing? Is it an Apple thing or?
No, it's at some point I like signed on for something that was offered and it takes notes of meetings, which is great, but it doesn't ask like it asks permission, but it just decides to insert itself in my schedule. So every time I have a meeting, it decides to join in. I think I have to actively kill it.
do I
Who's
need to
the
boot
who's
it?
the
Yeah, I think I can there we go, remove. Get out.
bit?
Oh no, I don't want to report it. I just want to remove it. Okay, it's good then. Okay, so I think I don't know, I think we can probably start. Um see yeah, yeah. Is there any sort of formalities or anything that needs to happen beforehand?
Well, I mean, I guess it's clear that you've taken all the courses that you need for graduation, right? That's done. So is that right?
Yep, yep,
Okay. That's the main thing.
great.
Let me just, and everybody who's supposed to be at this meeting is here. I always, sorry about this. I always forget who's the, uh, computer is very slow. Anyway, go ahead and proceed. Sorry.
That
So
sounds good. Yeah.
we don't need the mentor for this, do we? God, I hope not.
That's what I was checking. I don't
Yeah.
think so. I think for the PhD, it's not needed.
Yeah, I looked on the IPN website. I'm 99% sure it says no mentor.
Yeah, I don't know why, but it's the fact that we need it for the master's and not for the PhD always confuses me.
Let me see.
What does it say? Two prior must be attended and chaired by your supervisor. So
Ah,
no one else is required to be here. Yeah, just your committee and your supervisor are required.
Excellent.
that's
And
good.
of course, everyone is welcome.
All right. You think, shall we start, Ed? I guess.
Yes, go
All
for
right. So Darian, thesis seminar, take it away.
All right.
it.
All right. Well, yeah, thanks everyone for tuning in, and special thanks to my family who's coming in really early in BC time to hear about cell biology for an hour. So yeah, I'm going to be talking today about some of my thesis work, which is entitled Oligodendrocyte Substrate Adhesions in Myelin Wrapping. Oh, and keyboard's not working. Okay, there we go. So just to give a little outline of what we'll talk about today, I'll give a general introduction where I talk about the kind of the three main background topics, which are myelin wrapping, cell adhesions, and Netrin-1. And I'll talk about the hypotheses and the rationale behind the studies that I'm going to be presenting. I'll talk for a little bit about some of the methods that I've been developing. So these are three different things related to automated image analysis, high throughput microcontact printing, and making a myelin atlas. And then I'll talk a little bit about some of the data that's going in my thesis related to oligodendrocytes and cell adhesions. So let's talk a little bit about myelination in the central nervous system. So myelin is a multi-layered sheath of membrane that is wrapped around neuronal axons. So it's something that provides a lot of metabolic and structural support to the neurons, but it also makes neurotransmission much faster and dramatically reduces the amount of energy required for neurons to communicate. So myelin was an evolutionary innovation that allowed us to evolve much more efficient brains, and it's been very important and has allowed us to do things like give PhD seminars. Oligodendrocytes are the cells in the central nervous system that produce myelin. So they do this, kind of got this little cartoon below, in a series of different steps. Initially, the oligodendrocyte precursor cells migrate and kind of spread out across different neuronal axons. They then start extending branches out, and these branches start to coil around the axons. At this point, the branches start to extend into these long sheets of flat membrane, and these can coil around the axons many times. And then finally, they kind of adhere at the ends, and you end up with mature myelin. So understanding the cell biology driving myelination is relevant for learning and plasticity because myelin actually changes shape to change the speeds and different properties about neuronal firing. But it's also very important for understanding white matter diseases like multiple sclerosis. In MS, patients lose myelin, and there are no therapies that exist to protect or regenerate it. And the myelin that is regenerated by your body is usually made up of much shorter and thinner sheaths that don't seem to work so well. So we're really interested in understanding the cell biology here so we can try to find therapies for this kind of thing. So there was always a big mystery on the subject of understanding myelin, of how it actually forms. So this right here is a cross section with an axon in the middle and a myelin sheath around it. And kind of in the zoomed in section here, you can see that the sheath is made up of many of these different layers of membrane. And the important thing is these layers are extremely compacted. They're really just a few molecules thick. And so the question then is, how does this actually form? You have this kind of bulge on the inside called the inner tongue. That would have been like the leading edge that's traveling around it. But just like with humans, where we need muscles and bones to be able to move, a cell needs a cytoskeleton to be able to push forward and advance. But this space is just too small to fit much cytoskeleton. So this was a big question of how this actually forms in the first place. And it was really only kind of figured out in the last decade or so. So there was this paper in 2014, which kind of shows here their model. So this is an oligodendrocyte wrapping an axon, and on this side, it shows what its process would look like if it was kind of growing out in a flat environment. So as the myelin wraps around it, the leading edge is kind of bulging out, and then it compacts behind that. So as it's coiling, it tucks under itself, this leading edge, and goes around and around. And the way it's able to do this without needing a cytoskeleton is by actually using kind of a hydrostatic pressure type thing. So as the outer layers compact, they squeeze the remaining liquid in these inner layers, and that propels it forward around and around until you end up with just many layers of compacted membrane and just a little kind of bulge with the leftover cytoplasm inside of it. And so adhesion is very important for this. It was shown that oligodendrocytes downregulate their adhesion molecules that they use to actually stick onto this axon so that this inner tongue can glide smoothly around. And so this was published relatively recently, and this is the current model, is that oligodendrocytes must de-adhere to be able to have this kind of pressure-based pushing of the inner tongue around the axon. But there's been a few problems with this model. So for one thing, it doesn't really account for the edges. So if it is pressure, like this is the situation that we think must be happening, is that it must actually adhere on the two edges to constrain the pressure so it forces things to wrap around. If it did not have adhesions on the two sides, then you'd think the pressure would actually just make it expand out laterally and it wouldn't be able to wrap around so efficiently. So it seems like you still would need some adhesion for this to work. And there's been a few knockout studies that have come out that seem to be supporting this, where different types of cell adhesion molecules have been mutated in oligodendrocytes, including CADM4, NCAD, and JAM2. And in all of these mutants, you get aberrant myelination, you get thinner sheaths, some different kinds of deformities, including what I'm describing, in which the inner tongue just kind of expands out the sides instead of wrapping around. So it seems like the role of adhesion in myelination is not really settled. Oligodendrocytes probably still do modulate their adhesions to the axon to control how they wrap them, but it seems like there's probably more regulation going on here, and that's what the body uses to choose where myelin's happening and maybe control things about the sheath lengths and sizes. So understanding how this is regulated may lead to therapies and will just generally improve our understanding of how myelin's working. So this brings up netrin. Netrin is a multifunctional myelin regulator. It's been studied for quite a few years in the context of myelination. Netrin-1 is a secreted protein that's produced both by oligodendrocytes and neurons in the nervous system, as well as several other cell types. It has two main receptors in oligodendrocytes, DCC and UNC5B. So, and at early stages, when oligodendrocytes are still at that very young point, when they're kind of migrating around and just starting to branch out, netrin actually has a repulsive role. So this is kind of shown here. It's just a normal cell. You can see some nice good branches and things, but after treating with netrin-1, it has much smaller projections. But as the cells get a little bit older, you get this switch where netrin begins promoting growth instead. So this is an older cell at day 4. You can see some branching, but after receiving netrin, it gets much more branchy and generally gnarly. So this switch from repulsion to outgrowth suggests that netrin may be actually promoting myelination at later stages. And there's been some other research from other students in our lab and others that have shown that netrin's also doing things like recruiting mitochondria and shifting the energy metabolism of the cell and causing the expression of some myelin-related proteins. So we also see, looking in adult mice using conditional deletions of netrin as well as its receptors, that we get different kinds of defects at paranodes. So paranodes are at the ends of the myelin sheath. The sheath kind of has to curve down and all these different layers of membrane have to stick to the axon. So this is what they look like normally. You get these pretty nice loops, and this is a control cell here where you can see different paranodal loops all nicely adhered to the axon. But when we have netrin deletions or its receptors, we get things like loops detaching, loops becoming everted, and we lose the dense band of protein, which is holding them onto the axon. So we can kind of see this here with all of these disorganized and kind of fragmented paranodes. So this is really saying that netrin is doing something involved with the adhesion of the oligodendrocyte to the axon. This dysregulated paranode resembles the kinds of disorganization you have in things like multiple sclerosis and other leukodystrophies. Seems like netrin is controlling adhesion in some way that's important for myelin. So again, just to reiterate, netrin early on, it has a repulsive role where it repels OPCs and prevents them from growing. At later stages, it promotes paranode stability. And in the middle, we're still not quite sure. It's promoting expression. It seems to be doing things related to adhesion and growth, but we want to really find out what exactly it's doing for myelin wrapping and if it's participating in myelination. So how do we study myelination in vitro? So we use an old protocol published by McCarthy and DeVillis in the 80s. This is essentially, we take neonatal rat cortices, we dissociate the cells, we end up with a mix of a bunch of different glia. We put these in flasks and culture them for a few weeks in which they expand. And then at that point, we can specifically isolate the oligodendrocytes from that culture and then seed those. So when we seed these oligodendrocytes, we can grow them on a regular flat culture dish like we would anything else. But the problem there is we can't really study myelin wrapping. There's no axons or anything for the oligodendrocytes to actually wrap up. So they just grow a little bit, and we can't really look at these more specific processes. People have cultured them on neurons or cerebellar slices and things like that, but these cultures are very slow and expensive and technically challenging. So it's not really a great solution for doing things like screening for drugs and digging really into specific cell intrinsic mechanisms. So instead, there is this recent, about 10 years ago, it was published that you can grow oligodendrocytes on little tiny plastic fibers. So these fibers are produced by a process called electrospinning, where essentially you can just produce little tiny plastic strings of whatever size you want. So this company's been producing these that are about 2 microns in diameter, made of polyl-lactic acid. It's been shown that when oligodendrocytes wrap these up, this is an image of mine, you get long sheaths of myelin basic protein. So this is a protein that's highly expressed in myelin that I've stained in red here. So you can see these long sheaths covering the fibers in blue. Another lab has done electron microscopy and shown that you do get what looks like really bona fide compact myelin forming around the fibers. And yeah, here's a video of mine showing a cell just growing on it, if it's gonna play. Okay. Okay, whatever. So the fiber cultures are ensheathed the same way that axons are as oligodendrocytes differentiate. So this is a way that we can model actual myelin wrapping. And yeah, it has a number of advantages. Again, you don't have to use axons, so you can look at the specific cell biology of the oligodendrocyte without worrying about confounds there. They're very optically accessible, and they're amenable to putting different drugs on to see how that affects myelination. And they can be coded to see how different substrates affect myelination. So, so yeah, what does that mean, coding? So in the body, the cells inhabit extracellular matrix. So this is basically a jello-like environment full of these big positively charged proteins that the cells can kind of grab onto with their negatively charged plasma membrane. And this allows them to move and take shape. So proteins like netrin-1 can also be embedded in the extracellular matrix, and this allows them to serve as kind of guideposts. So as cells are migrating or moving around, they can encounter these different cues and then respond. So in vitro, we need to give them some sort of a matrix to hold on to as well. And we do this with an artificial polymer called poly-D-lysine. So this is made of repeated lysine residues, and these are characterized by having these positive amine groups sticking out. So when we take poly-D-lysine and we put it on a glass surface or something, the poly-D-lysine sticks to the glass and presents all of these little positive charges to the cells, so the cells can happily grow on it and adhere. We can also do the opposite, and we can use things like polyethylene glycol. And this is kind of the anti-PDL. It's very slippery and cells cannot grab onto it. It's kind of negatively charged. It's been used for things like preventing biofouling. Milk companies coat milk cartons with this to prevent bacteria from sticking to the sides of it. And similarly, mammalian cells cannot grab onto it. So you can actually get polyethylene glycol that's been grafted to PDL or PLL, which is very similar. And so in that way, we take this big positively charged molecule and we stick these negative slippery ones onto it. And so then when we coat these onto glass, we can have the following kind of situation. Regularly, glass is negative and cells cannot adhere to it. But as I said, when we take PDL, we can have this positive molecule stick to the negative glass on one end and present positive residues to the cell to adhere on the other end. And when we have this PEG, we can have an even more complicated scenario where we have the positive PLL sticking to the glass, but then presenting the negative PEG to the cell. So once again, the cell cannot stick. So by using these different coatings, we can control where cells are adhering and how much they can adhere to their substrate. And there's one other polymer that I want to talk about, which is dendritic polyglycerol amine. So this is a relatively newly discovered or created molecule. It's a type of dendrimer. And so dendro is from the Greek word for branched. As an aside, that's also where oligodendrocytes are from. It means few branched cells. But so in this case, dendrimer is referring to the branching of the molecule. And so it's, rather than being a linear thing like poly-D-lysine, it's kind of this ball or snowflake shape that's surrounded by positively charged residues. So this one in particular, DPGA, is one that has been characterized a little bit as a cell coating, cell substrate coating. The idea is, yeah, all these amine groups on the outside will act similar to PDL, but it has a much higher charge density and it's more stable. It's less likely to break down than these linear molecules. So it can maybe give us a more powerful and more stable way of coating our cell culture dishes than PDL. So it's been used to study neuronal cultures in a few different studies, and they found pretty impressive results where it's promoting much more cells growing in general, better cell survival, and more long-term cell viability using multiple different iPSC lines as well as primary neurons. And so our question is this better for oligodendrocytes as well? So what are the hypotheses of this work? So there's two main things. Firstly, we can see from the literature that some adhesion seems to be required for optimal wrapping to constrain lateral growth. So that's the first hypothesis, is that it's not that you have no adhesions to get best wrapping, rather you do need a balance between positive and negative adhesion. And relatedly, we can ask, do sheath lengths depend on the axon stickiness? And is this a way that axons might control myelin that's being deposited on them? We also have the question of, does Netrin-1 promote process outgrowth in these early two-dimensional oligodendrocyte cultures and stabilize sheaths in we know that Netrin-1 promotes process outgrowth in 2D cultures and stabilizes sheaths in aged mice. So we can ask, does it regulate sheath wrapping as well by increasing growth or adhesions? I should mention also, I've got a little featuring some AI images in this, so you can see if you can spot them. This is a cyberpunk style oligodendrocyte. So there's going to be a few more of those. So I've got three aims then. Firstly, it's to determine how substrate adhesions affect myelination. Secondly, to see how Netrin-1 is regulating wrapping. And thirdly, in general, to develop high throughput microscopy-based methods to help study myelin biology. So let's talk a little bit about some of the methods. So when we have these electrospun nanofiber cultures, we end up with, if we image a whole well, something like this. So we have all of this in red, it's the myelin basic protein. So these are the myelin sheaths that the cells are depositing on the little electrospun fibers. If we kind of zoom in, we can see something like this. We just have all of these red streaks responding to or corresponding to the sheaths. So this kind of thing, these cultures are pretty easy to scale up. You have them in 96 well plates. You can do pretty high throughput experiments, but the problem is analyzing this is difficult. You need a person to come around and individually trace these sheaths. And the hard part is deciding which sheath belongs to which cell. So what we'd really like to do is be able to measure per cell, how is the myelination changing when we put drugs on and things like that. But yeah, the big bottleneck is having a human analyze it. So to solve this problem, we had a little collaboration with Jack Antel's lab and Doug Arnold's lab to create an AI basically that would go around and do this analysis for us. So this was based on a 2015 archive paper from Ronneberger et al. This was back in the really early days of AI. It was kind of the Stone Age relative to where we are now. But they used this UNet architecture and they were able to get really high quality segmentations of cells from just DIC images using this. So yeah, with our help from Rob Brown from Doug Arnold's lab, Tiger Shu and I were able to adapt this UNet architecture to be able to trace oligodendrocytes instead. This is just kind of showing what some of the different layers of the network are seeing and the kind of features that they're responding to. So what we end up with is it's not only tracing the sheaths that it finds, but also doing it specifically for one cell at a time. So in this case, it decided that these sheaths are one cell, and it's probably going to decide that these sheaths belong to another cell. So to validate this, we had two experts who had traced this kind of culture before analyze it, as well as a couple of undergraduate summer students analyze a series of images and then the algorithm, and we compared how close all of their traces matched. And what we found was that comparing the two experts with the network, they were all pretty much indistinguishable, whereas the undergraduates performed significantly worse. So we counted that as kind of meeting relatively the gold standard, so that was good enough. So this it seemed like it was doing a pretty accurate job. And so we started using this, and so we used this and for one additional validation, which was to look at how laminin coatings were affecting myelination on fibers. So this was something that was published on nanofibers recently. And in this paper, they showed that there was a increase in the number of sheaths per cell. So this was published in the paper, or sorry, this was in the paper, and then we found the same thing. But because our algorithm was allowing us to analyze hundreds or thousands of cells, we had a really high N, and we were also able to pick up a subtle change in the sheath lengths, and that sheaths were getting slightly longer on the laminin fibers. We've also used this since to be able to optimize our cell culture. So we've screened different medias and different isolation methods to find the most optimal. This was made much easier with the algorithm. And it's since been used in 3 different papers looking at iPSCs as well as ways of overcoming remyelination blocks in multiple sclerosis. So the second method I'll talk about is microcontact printing. So microcontact printing is a technique where you can literally just stamp a molecule, such as a protein like netrin, onto a substrate. And you do this using a polydimethylsiloxane stamp or PDMS stamp. So this is something, this is an example of mine. Here we just have different dots of netrin that have been deposited, and we have some different epithelial cells that are growing on it. And we can see already that different cells respond very differently when they encounter this kind of substrate-bound Q. So the netrin is, you can see this cell is really avoiding it, whereas others seem to be really liking it. So this type of technique allows us to see how cells interact with an immobilized protein. As I mentioned in the body, the extracellular matrix sometimes holds on to these proteins and kind of presents it so that cells can encounter it. And we can kind of mimic that in this way by depositing a dot or a stamp of something and seeing how cells respond. So this was used in 2014 by Sebastian Ricot, an old student in our lab. And so he was looking at how cells are responding to netrin, and he came up with an important concept related to this, which is the topic of the reference surface. So this, in this case, we have netrin is in green stripes, and we have, we can see that in most cases cells are, sorry, at the cases like these, the cells in red are really growing on the netrin stripes. But over here, the netrin is, or the cells are not, they kind of don't really care about the stripes. And the difference is here we have a very positive, very sticky background, whereas here we have a very slippery background. So this makes sense. The cells have a choice. They're growing on either something they're really stuck to or something that they're not really stuck to. And then they encounter a cue like netrin. So if they're really stuck, they won't be able to respond. But if they're at the right kind of background stickiness, then you will see a large response to the cue. So tuning the reference surface is an important way to study how cells are interacting with these kinds of surfaces. And it also shows how like changing the adhesive background can change a lot of things about how your cells are behaving. And so he did this by using different ratios of PDL and PEG. So again, that's this positive and kind of negatively charged things. And yeah, just has different ratios from fully sticky to fully slippery and everything in between. So I wanted to be able to use this, but it's difficult to do it on cover slips, which is kind of what it was traditionally done on. So these are little glass pieces that you can culture cells on. Problem is this requires a lot of cells. It's very low throughput. And I wanted to be able to use multi-well plates, which are much more efficient and automatable and use far fewer cells. So I developed a method using these biopsy pens, which are essentially like a little kind of hole punch pen. This allows you to punch out little pieces of stamps, which you can then ink with the protein solution, dry off, and then deposit in wells. And so this way you can, in a much more high throughput way, deposit patterns in a multi-well plate, and you can study how cells are interacting. So here's just an example in one of these 96 well plates showing cells responding to kind of a honeycomb pattern. And we've also been able to do this on the nanofibers. So this in cyan here, you can see the fibers, and in red, you can see a rhodamine that was printed on top. So the final method I'll talk about is a little bit of a deviation, not so much talking about cell biology here, but this has been a project to map the marmoset myelin using a multimodal MRI as well as histology. So this is addressing a problem. Marmosets are becoming a much more commonly used model organism in neuroscience research. They're a small non-human primate that are much more easy to take care of than other non-human primates, but they exhibit complex social behaviors and kind of higher order functioning than mice. So they're a very powerful tool to be able to study things like autism and social factors and other kinds of neuropsychological issues. So the problem, though, is that there isn't a lot of tools right now for researchers who want to study in marmosets. So there's a number of atlases that have been made showing the brain morphology and different cell types, but they have a lot of limitations. Like for one, they've all been using neuronal and nuclear histochemical stains, so there isn't actually any resources to look at glia or myelin. Most of them are based on MRI techniques, so in those cases, they're confounded by tissue microstructure and iron content. There's a greater need to validate some of these atlases with histology. And finally, none of these are using fluorescence microscopy. So there's no multiplexing. They're always just staining a single factor at a time, which prevents you from looking at how different components or compartments are interrelated. So in collaboration with the Tardif lab, who are a marmoset MRI lab, we set out to build a atlas that was going to focus more on myelin, but also use fluorescence imaging to be able to label multiple different cell types at the same time and give a really rich atlas that will be useful for the field. So the production of this atlas is still ongoing. We've done a lot of the microscopy, and the MRI is currently being correlated with that. So there's just a little clip from what's going on there. In all, we've imaged over 200 brain sections stained with 5 different markers, and all automatically imaged and processed. And yeah, we're building this atlas now. In the meantime, though, we've published the protocol on the batch staining method that we came up with to be able to uniformly stain this huge number of slides. This kind of thing is usually done with histochemical stains, not so much with immunofluorescence. So we had to adapt a few things to be able to do this in a reproducible manner. But yeah, we recently published this high throughput staining pipeline, as well as a set of these 5 different antibodies that were validated for use in marmoset tissue. Yeah, then this has just recently been, sorry, not published, but submitted, and we'll stay tuned for the atlas to be made available, but that will be a great open access tool, I think, for the community. So to talk a little bit about the data now. So as I was saying, the main interest here is manipulating non-specific substrate adhesiveness. Like we want to be able to control how much the cells are sticking to what they're growing on without triggering specific signaling cascades. And so again, we're going to do this by taking the poly-D-lysine and the polyethylene glycol and using a few different ratios.
PhD committee in about an hour.
Yeah, so we'll do a few different ratios of the PEG and PDL. And in addition, I'm testing the DPGA. So to first just see if these coatings were going to work, I just took a random cell line, HEC293 cells, and just grew them on glass cover slips coated with these different ratios to see if we could actually prevent the cells from adhering. And so this is kind of what we saw. At this end, we have the PDL coated cover slips, so these are very sticky. At this end, we have the PEG ones, so they're not sticky, and we can see there's almost nothing stuck to them. And then with DPGA, we had pretty healthy growth as well. So quantifying the cell counts, we see that there isn't much of a difference as long as we have at least 50% PDL. Once we get into lower amounts of PDL, higher amounts of PEG, we have quite a bit fewer cells and almost none adhered when it's pure PEG, and DPG has about the same cell count. Looking at the morphology, we can see that it's having some effects. This is kind of what HEC cells look like normally. They see some nice little branches and filopodia coming out. On the half PDL, half PEG surface, you see less processes coming out, likely because they're not able to adhere to as many things. On 100% PEG, again, there was almost no cells. The only cells I did see were these miserable kind of balled up ones. They ball up because they can't adhere to the plate, so instead they just stick to each other. And on DPGA, though, we saw much larger and more healthy looking HEC cells, or at least more spread out HEC cells. So I also quantified cell clustering, and I did not include the 100% PEG in this case because everything was clustered. But just looking at the degree of nuclear clustering, we actually see that we have more in the DPGA case, even though there's about the same number of cells overall. So not sure how to interpret it, but it does show that DPGA is active and potentially doing something different from PDL, but ultimately it's still an adhesive substrate that the cells are able to grow on well. So now we wanted to test this on the nanofibers and see how this was going to actually affect myelin wrapping. So here we coated fibers with the same ratios of PDL and PEG or DPGA. So the exact same treatments here, but here we're doing it on the nanofibers and looking at how oligodendrocytes are able to wrap them. And so this is kind of what we get. So on top, I've got the nanofibers and cell bodies in blue and myelin in red. On the bottom, it's just the myelin basic protein. So you can see generally, again, they look okay as long as there's some PDL, but there's a big reduction at the PEG level. You have far fewer cells, and I'm not sure how clear it is, but rather than having these straight, thick sheaths, which are wrapping the fibers, we have a lot of these really thin kind of winding processes. So here they're not actually wrapping, they're just growing along top the fibers. And in DPGA, we had some very nice, healthy, thick ones. So I want to quantify this, looked both at the average sheath length for each cell and the number of sheaths that each cell had. Oh, sorry, before that, I looked at the proportion of ensheathing oligodendrocytes. And so here, yeah, we see that it's about the same for all the conditions. It's just a little bit less, or a fair bit less in the PEG group. So that one is significantly different from all the others. The others are relatively unchanged. But yeah, looking at the actual lengths of the sheaths and the number of sheaths per cells here, we could see that there's quite a big difference. So we have significantly more sheaths when we have the 25% PDL condition. So the less sticky fibers are getting more myelin, but we're getting kind of the opposite trend with sheath numbers, right? So we're going up as we get less sticky, and then we're going down in terms of lengths, and we're going down in terms of counts. So what's happening is the oligodendrocytes are producing roughly the same amount of myelin in all cases. It's just when they're on a sticky fiber, they end up with a lot of small sheaths, and on a slippery fiber, they end up with fewer long sheaths. In the PEG group, interestingly, so yeah, there were very, very few cells that were able to myelinate, but the ones that were able to wrap looked fairly similar. The lengths were comparable to just PDL-coated fibers. So it seems like once they were able to start wrapping, they were able to continue. But yeah, there were many fewer wraps per cell. So not only were fewer cells able to wrap the fibers, but the cells that were wrapping had fewer processes that were able to do it. And the other thing is that DPGA was, despite we think being a very sticky molecule, it had also very long sheaths on it, and it had much less of a reduction in the number of sheaths. So DPGA actually had the most myelin overall, which was interesting. So the last thing just to point out here is, yeah, with the PEG, as I said, they're not really able to grab onto the fibers. So this is an example. Here's just a PDL. We see a lot of growth on the fibers. These are, again, MBP positive sheaths. Here we just see some kind of sad little growth along the top here. But if we look below them on the bottom of the plate, these fibers are suspended about 10 microns above the base. We can see often in the PEG case that the oligodendrocyte kind of slipped through the fibers and grew on the bottom instead. So here the cell was mostly growing on the fibers, and it just has a little bit of growth below. But here, yeah, the cell is mostly growing below and has just managed to hang on a little bit to these slippery fibers. So it really just goes to show that the PEG is not preventing the oligos from growing or surviving, but it is preventing them from wrapping. Yeah, so that's what we got from the adhesion studies. So I'll come back to this a little bit later. But let's talk a little bit about netrin now. So that's kind of looking at the non-specific adhesion, but again, netrin we think might be inducing adhesions. It might be making the cells adhere. And part of why we think this is based on the localization that we see in vivo. So this is from an old paper from Andrew Jarjor, and this is staining netrin in green here. This is looking, there would be an axon in the middle and myelin around it. So we can see that the netrin is really localized right between the axon and the myelin, as if it's there to instruct the myelin to do something. We also see it's located at paranodes, and this is at the ends of the myelin sheaths where they need to be adhered, and this is where we see the defects when we mutate netrin. So we want to see again, yeah, is netrin then promoting adhesion? In that case, we might see sheaths are not growing larger, or is it promoting myelin extension? In that case, we would see more sheaths. So to mimic this, we wanted to just coat the fibers with netrin. So we took recombinant chick netrin 1 and we just absorbed it onto the fibers the way we would any of the other polycationic proteins. And this recombinant netrin 1 has a MIC tag. So here I've stained it MIC in white. So this is fibers not coated with netrin, and this is fibers coated with netrin. And you can see that even after 14 days in culture, the fibers are still labeled. So we are able to adsorb netrin onto these fibers, and then we can see how the cells are responding. So when we did the experiments, we pretty immediately noticed that this was not the right strategy to look at this. There was far fewer cells in the netrin 1 condition than the PDL. When we actually counted the cells using the automated AI system, we got about an 85% reduction in the number of MVP positive cells that were ensheathing in the netrin 1 case. So as I mentioned in the beginning, netrin 1 has a repulsive role early on, so this makes a lot of sense. When we're seeding the cells at these immature states, they're still being repelled by netrin, and they're just landing on a sea of netrin, so they're not able to adhere to anything. Many of them are probably dying, or at least failing to differentiate. So we think this is what's happening here and what's causing this total lack of myelination. But the interesting thing is when we look, oh, sorry, and I'll just say this is also interesting because in multiple sclerosis, you actually have netrin fragments in the lesions that are left. So this has been postulated to be part of why you get poor remyelination in multiple sclerosis is because you have netrin and perhaps other repulsive factors in the lesion. So by coating the fibers with netrin and then trying to get them to be myelinated, we kind of are modeling an MS lesion environment. So it's not useful to see how netrin's affecting wrapping, but it is useful potentially as a model for MS lesions. So I'll come back to that a little bit later as well. Anyway, so we wanted to see though, of the cells, of this like 15% or so of cells that are able to myelinate in the netrin condition, how do they look? Are they surviving? Are they looking better than the cells in PDL? So with this, we again use the power of UNet, even though we only had a very small number of cells on the netrin condition, we were able to analyze a few hundred wells and get enough cells to do some statistics. So yeah, looking at the mean fiber length per cell, we do see that we have lower sheath lengths in the netrin group. However, it's not very different. I was really expecting to see a massive reduction based on how much less myelin there was overall. But yeah, the remaining cells that were myelinating were looking pretty similar. So we looked at a number of other different metrics of sheath growth. So this is MVP intensity, total sheath length, coverage of the cells, things like that. And again, generally, there wasn't a lot of difference. Netrin is in orange, PDL is in blue. Most of the distributions are fairly similar. But we did notice that there was a subset of the netrin cells that were actually larger. Even though the means were generally lower, they were having longer sheaths, longer or larger total coverage and higher MVP intensities. So it seems like there might be a subset of cells that are responding positively, but we weren't sure if this was that they're just, once they're able to get past the initial repulsion, all cells will respond positively, or if there's perhaps just a subset of oligodendrocytes that respond to netrin. So to look further at this, we changed strategies. Instead of coating the fibers with netrin before seeding cells, we thought, what if we put netrin on at various time points as the cells are growing? So we can kind of target these different stages. So here I'm putting netrin on at 4 different time points, like when they're in the kind of precursor stage and they're still migrating around, when they're starting to send branches out, when they're starting to wrap, and then when they kind of should be finished wrapping. So initially, looking at these different time points, we did see that we had a drop in the number of oligodendrocytes with at least 3 sheaths early on, as expected when it was repulsive. But putting it on later, we saw that we actually had more oligodendrocytes with at least 3 sheaths. So this did seem to say that at later stages, oligodendrocytes might be responding more positively. So we wanted to look at the morphology per cell again. And so looking here at the average sheath lengths, yeah, we see that they are generally reduced on the first few days, but when we put netrin on later, they go back up to baseline. So only the young oligodendrocytes are getting this negative response to netrin. When we look at the sheath area, interestingly, so this is the total pixel count, we actually see we have more when netrin's going on at day 4. So right when they're in this middle of the wrapping stage, if we put netrin on at that point, we actually get thicker sheaths, it seems. And looking at MVP intensity, we didn't really see a difference at the later time points, but we did have higher intensity when we put it early on. And this just seems to be because oligodendrocytes are producing a somewhat constant amount of MVP and the sheaths are forced to be smaller. So it seems like substrate-bound netrin-1 potentially could be increasing outgrowth. So for this, now we wanted to look a little bit closer. We wanted to get around the issue that it might have to be bound to a substrate. In the previous experiment, we were just adding netrin to the culture wells, so the cells were surrounded by it. They didn't get any sort of directional cue to it. They wasn't really telling them to adhere to something. Rather, they were just receiving signaling from everywhere. So for this, we wanted to use microcontact printing instead to be able to get a nice stripe of netrin and see exactly what happens when a cell encounters it. So I used the microcontact printing protocol I mentioned to stamp stripes in these 96 well plates. So this was just a 2-dimensional culture, not on the nanofibers now. This is, again, just looking at some random endothelial cells that made it into the culture. These cells really like netrin, and we can see that they're growing right on top of the stripes. So we have this kind of little internal control showing that the procedure's working. And what I also saw as I started looking was a lot of cells that kind of stopped on the stripe. They were able to grow out in other directions, but on the side that was encountering a stripe, they stopped. So this really did seem in line with the idea that netrin is inducing adhesion, that it might be, these processes are growing out and then they're stopping when they encounter it. So I wanted to quantify then the number of cells that were stalled when they encountered the fibers or these stripes. And we've got three different conditions here. There's the netrin stripes, there's PDL stripes. So this is a positive control, it's just something sticky. And then we also have some PEG stripes. And the background here is also, sorry, 50% PEG. The background is also 50% PEG. So this should do nothing. Essentially, I'm stamping the same thing that everything is coated with. So it should do nothing. So what we saw looking at the number of cells that were stalled was that there was about 20% of cells were stalled like this when they encountered netrin or PDL stripes, whereas only about 6% in the PEG case. So we did see more cells that were stalling, but not as much as expected. There were still most of the cells were able to grow past it. And interestingly, when I looked a little bit more at some of these cells, I saw a lot of them actually grew very far past of it. So you saw this kind of thing where it's, growing out a little bit in all directions, but on the portion that's contacting the stripe, we're getting big growth coming out. So I wanted to quantify then, are the processes that are contacting the stripes growing more than the other cell processes? So the way I did that was tracing a convex hull around the cell, soma, and stripe, and then partitioning them into on and off stripe. So I had a cell like this, and I would trace this hull around it, and then I would mark which angle, the angle of the processes that are growing onto the stripes. And then by doing this, I was able to multiply the length or area measurements on the stripe sides by this angle to essentially normalize it and be able to compare the growth off versus on stripe. So looking at the PEG group, so again, this is a negative control. There should be no effect from the stripe. We expect the on-stripe processes to grow just as much as the off-stripe. We do see that. There's no difference when we look at the total pixel area covered, and there's no difference when we look at the integrated density of phalloidin here. So it seems, yeah, like the PEG stripes are not actually interfering with anything. We see that here. They're just kind of growing out in all directions. When we look, however, at netrin, we see a little bit of a difference. So we do see that the stripe processes in many cases are getting much larger. They're covering a much larger area, and we have a much higher integrated density or like intensity in them. So we can kind of see that here, that this process gets very large and bright. In PDL, we saw a little bit of the same trend where processes growing onto stripes were a little bit larger and a little bit further growing, but it was not significant. So it seems like, yeah, netrin maybe is inducing some more growth, but something else interesting is happening. So you can kind of see here that this cell has a lot more red signal. This is MVP again. And I saw this repeatedly. It seems like a lot of the cells growing on netrin fibers seem to have more myelin basic protein. So I wanted to, again, look a little bit further and really look and see, are we seeing more signal intensity like on the stripe or after the stripe when we have netrin compared to PDL or PEG? And so looking at that here, we do in fact see this is the kind of the before stripe compartment, on stripe compartment, and after stripe compartment. This is for PEG. We have what we would expect, but there's kind of less and less material as you get further away from the body. In netrin, though, we do see we actually have more material on the stripe and more after the stripe than we do beforehand. And looking at PDL, we have another different case where we have most of the growth on the stripe and then less after it because it's being stalled. So I'm still kind of sorting out the best way to handle this statistically, but I think this is interesting results that are really showing that netrin does seem to be promoting growth only on the process that's contacting the stripe. But as I said, it seemed like MVP levels were going up as well. So I quantified the MVP intensity, and I did see that there was a pretty significant increase only in the netrin groups, not in the PDL. So together, this is saying that netrin does seem to be promoting more outgrowth of these processes that are contacting it, and also that netrin is causing more MVP expression in the whole cell. So finally, the kind of magnum opus to work towards is, are we able to do this microcontact printing on fibers, and how will that affect actual wrapping? So we know again that two-dimensional growth is not the same as fiber wrapping. If we have these netrin stripes, are we going to see that the cells are going to stick and adhere on them when they encounter them, or is this going to incentivize them to grow past it? So are we going to see something like this where the cell's just growing kind of up into the stripes and then not past them, or will it grow further? And so what we see is that it seems to be growing up into the stripes. So we see this type of thing here where we have cells that are often in the middle of these bands of stripes, and they grow until they reach them. We have some more of these here. It's a little bit less clear. And so this is just zoomed in here. You can really see how these different processes have grown until they've encountered the green stripe of netrin. So I'm still working on quantifying this, but I think these are really encouraging results that are showing that netrin might be inducing adhesions at these ends. And the interesting question will be as to whether it's also promoting more efficiently wrapped sheaths in between it, which is what we would predict if these adhesions at the lateral ends are required. So let's talk about the conclusions today. So yeah, this image I really like. This was brain cells studying brain cells in a pop art style using the Microsoft Designer AI. So we saw that low adhesion results in some of the longest sheaths, but that some adhesion is necessary for wrapping. So this raises the question, is adhesion only required at presumptive paranodes to constrain wrapping? Like, do you need any adhesion in the middle of the internode to wrap? And so this is something that could be addressed in the future with microcontact printing. So we also found that DPGA elicits longer sheaths and more myelin overall than any of the other coatings. So this raises the question of how exactly is it doing this? It seems like it might be eliciting some sort of a biological response, but that will have to be followed up on. We found that substrate-bound netrin-1 prevents most oligodendrocytes from myelinating fibers, which raises the point that netrin-1 could, because it's found in MS plaques, this could be a way of studying the block of remyelination that happens in MS. And finally, we found that in mature oligodendrocytes, netrin-1 induces higher MBP levels, wider sheaths, and process expansion in 2D. And this is all pointing towards netrin-1 inducing some sort of a myelin wrapping program. So to talk a little bit more about the netrin side of things, it's clear that netrin is playing a role in myelination, but it is much more complex than what we initially thought, which is that it's just promoting adhesion or growth. Again, we do see it increases MBP and branching. We've got other studies showing that it promotes like myelin levels of MBP biochemically and things like that, but we do not see that it's increasing sheath lengths. And this is an interesting contrast with some of the other stuff in the literature showing that other factors like laminins and ephrins do increase 2D outgrowth and also increase sheath lengths. So netrin seems to be maybe targeting a different mechanism involving wrapping than some of these other factors that have been studied. But it's interesting, and we should really be looking into the difference. We saw that netrin-1 triggers both local and global effects. So looking at the microcontact printing data, oligodendrocyte processes that encountered netrin grew longer, but the MBP in the whole cell was elevated. And this parallels what was seen by Diane Nakamura, a former student, who showed that when oligodendrocytes contact netrin, you get local recruitment of mitochondria at that spot, but the overall cell metabolism is shifted. So it seems like netrin is doing things locally. It maybe is inducing certain sheaths to myelinate, but it's also triggering changes in the cell body that are affecting the entire cell. And we also saw that oligodendrocytes are responding very heterogeneously. And so this is something I would have loved to have dig into a little bit more. But through many different experiments, we saw only a subset of oligos were responding to netrin, and I was never able to really find a morphological criteria that told us whether a cell would respond or not. So we know now that there's a lot of different oligodendrocyte subtypes and that these seem to have different functional properties. So I think a really important follow-up will be seeing which ones are responding to netrin and what does that actually mean for the physiology. We saw that DPGA promotes in vitro myelination. So yeah, again, we saw longer sheaths and more myelin per cell than with any of the non-specific coatings. So this really suggests that it might be engaging some sort of specific signaling. One idea is that DPGA could be activating a specific receptor on the oligodendrocytes, but it might also be binding secreted extracellular matrix molecules. So for example, heparan sulfate proteoglycans are known to promote myelination, and they're positively, or they have negatively charged groups that could be interacting with the positively charged dendrimer. So it's potentially, we have like a layer of extracellular matrix being deposited that dendrimer is really, stably storing, that are then helping the cells to wrap. And it's important to follow up on the signaling to understand it, because this has been some pretty strikingly positive results on myelination, and this kind of thing could yield drug targets for multiple sclerosis. We saw that, it also raises the point that DPGA could be a useful tool for things like nerve regeneration scaffolds or other devices. We don't necessarily want oligodendrocytes wrapping up a nerve scaffold, but the fact that it seems to promote their survival and ensheathment might mean that they will be more likely to wrap up recovering nerves. And finally, as it did for neurons, DPGA could open up new long-term cell culture options for oligodendrocytes, especially iPSC-derived ones that need to be cultured for much longer than rodent cells. And related to this, as longtime viewers are aware, the nanofibers that I've done most of these experiments on are no longer commercially available. There are a couple of other companies that produce them, but I've tried them, and myself and others have had a lot of issues with cells not actually myelinating them. These companies are producing fibers that are smaller in different compositions. We're not sure what exactly the difference is, but they're just not being wrapped well. But I tried one of these with dendrimer, and I saw that, so this was kind of a collagen nanofiber produced by this one company. When the collagen's uncoated, which the company advises you to do, again, you just get these really thin growths along the fibers. I don't have a way of labeling the fibers here, but you can see these linear segments that are growing along them, but nothing that looked like actual wrapping. Whereas when I coated the cultures with DPGA, I saw a number of cells like this, where we had, zooming in here, actual thick sheaths that you could see. This is what they look like when they're really wrapping a fiber. So it seems like DPGA might be the secret to make these kind of suboptimal nanofiber devices usable for doing these kind of myelination assays. So that would be really, really great, I think, for the whole field. And the final thing to mention is just, the overarching goal I've had has been really to develop tools that can accelerate research into myelin pathology. And I think we've addressed that in a number of ways. So firstly, by creating open access image software, both the AI as well as some of the other software in MATLAB and Python that was written for this, again, have been used in several other publications and helped my own work a lot. We're at a stage now where we have automated liquid handlers and microscopes that allow you to do very high throughput experiments, but the bottleneck is still often analysis. So I think by making these types of tools available, we can really help speed up research in the myelin field as well as all other areas. There is the discovery that by coating netrin onto these fibers, we can kind of model the MS lesion environment. So this could be a very powerful tool for screening. Right now, when we're studying remyelination, it's usually done in animals, and this makes it very, very slow and costly and impractical. But by being able to deposit proteins like netrin-1 or even just MS plaque directly on these fibers, we can study how cells are interacting with it, see why they're unable to myelinate in the MS lesion environment, and try to overcome it with different drugs. So I think this is something that should be followed up on and is potentially an important finding. And finally, the creation of high-quality marmoset myelin atlases and the related methodology, I think, will help a range of things. But again, we're specifically looking at myelin-related disorders, and using marmoset's going to dramatically accelerate that kind of research. And by correlating the histology with MRI, as I was saying, that we can both refine MRI methods as well as, provide new tools for the community. So thank you very much to my lab. Special thanks to Jean and Nan, who helped with a lot of culture things and some of the marmoset data, and my very talented undergrad, Mavi, who did a lot of analysis help. Thanks to collaborators Tiger, Rob, and Chris, and my committee members, Jack and Ed, and the funding agencies. And thank you very much for listening.
Oh, really nice talk, Darren. Really, really nice. So Ed,
Yes, sir.
As a matter of protocol, do we keep the audience in for questions? I think we do.
I think we can. There's no reason not
Yeah.
to. This isn't a test necessarily, so I think it's more just a chance to. We will have a closed session at the end where we'll just decide whether there's enough to proceed.
Yeah.
But the audience can ask questions as well, of course.
Yeah. Great.
Can I start off with some questions, or
Please.
go first? I thought it was a great presentation, Darian. It highlighted how much you've done. It was really quite impressive. I always have this in vivo perspective, and I was wondering, there's a lot of focus on adhesion onto the axon or the axon-like substrate, and then you've clearly made provided some evidence that contrast may be more important than absolute adhesion in many cases. And I'm wondering,
right,
in vivo, are there circumstances where we should be looking not at signals from the axon, but at the degree of adhesiveness from the environment, from other oligodendrocytes, axon-axon, oligo signaling? Are there cases where that would be where what you've seen in vitro could actually tell us something about what we should be looking for in vivo?
Yeah, definitely. And I think this mostly comes back to extracellular matrix, and that's where we would see differences. So like I think I remember some studies showing that extracellular matrix was composition was different in MS lesions. So again, if we have different kinds of ECM coding axons as well as just in the environment, you'd think that would affect these kind of non-specific wrapping phenomena. So that's one big thing is, yeah, I think in pathologies when we have changes in ECM or as well as on like surface expression of different adhesion molecules on axons might be happening. And I think for demyelination, it's also important because so axons have oh, I'm blanking on it. I think adherins. There was one of the studies looking at mutating different adhesion molecules was mutating cadherin on the axonal side, and in that case, you also got these myelin defects. So when we have, I think axons might be presenting a kind of an optimal adhesive environment to the myelin when they're developing. But then after that, in adulthood, they might be downregulating these things or changing the adhesion molecules. And so then when they're demyelinated and a new oligo comes in, maybe we need to tell the axon to present the same adhesion molecules it had during development. So I think, yeah, I think the pathology context, I think there's a few areas where we should be thinking about adhesion.
Yeah, I mean, it sounds like good news because the extracellular matrix seems like a much more accessible target than trying to change, perhaps, than trying to change axonal expression or
Mm.
oligo. You might be able to just apply a drug and get immediate effects.
Yeah,
The
that's very true. Yeah. Yeah,
the other,
maybe just squirting dendrimer in. I'm just kidding, but
yeah, something like that would that would be amazing. But yeah, the other question had to do with this observation of young versus old. And again, the same kind of question. Do you think that's a feature rather than a bug? And if so, what are some examples where this kind of anti-unsheathing signal early on would be important, or how do you see that being important in setting up the initial myelination?
Yeah. I mean, I think it's interesting. Like it parallels what we see in axon guidance, where for a few different axon guidance systems, like Netrin's a good example. You have an attraction and then you have a switch and a repulsion. And so I think similarly, the repulsion, I mean, at an OPC stage, we know oligodendrocyte precursors are born ventrally and then they have to migrate dorsally in the spinal cord. So Netrin expression from the ventral cord is what guides them to move to the other side. But then as they mature, we want them to stop migrating and start myelinating. So if we have an intrinsic maturation program that switches this, then that would work out really nicely to get them where they need to be and then get them to start myelinating right away. There's also, yeah, like speaking more specifically at a sheath level, you would want, there are many neurons and parts of neurons that are not myelinated. So like dendrites and neuron cell bodies, for example. Mutating certain cell adhesion molecules like JAM2 does lead to myelin forming on dendrites and cell bodies. So we know that, yeah, certain cues presented by neurons are telling myelin to stay away. And so Netrin might be having that kind of role as well. It's possible that yeah, that it's being expressed in certain regions to keep young oligodendrocytes off of it. And just speaking temporally, I guess also because the oligodendrocytes are born a little bit later, the neurons want to wait until they've probably finished growing before myelination starts. So yeah, if they are expressing a lot of netrin as they're growing, they're going to be keeping the oligos away. But then if the oligos switch at the right time, then, if the oligo switch is synchronized with when the axon finishes growing, I guess you'd be set up to start myelinating right away when you need it.
Okay, no, that's really interesting. One last question I want. So there are these, I guess, paranode, or is it paranode or juxtaparanode, that the phenotypes that have been clearly reported late in life in the animals that have netrin deficits or netrin signaling deficits. And I was wondering, do you think that that may, rather than representing a maintenance issue, may represent remyelination issues? Is that how you're thinking about it, or could it be? Well,
Yeah,
I'm wondering why these effects are late, and whether that could be due to ongoing remyelination.
yeah, no, I think that's very likely. So there's been a few studies showing that, yeah, you do have continued myelin synthesis, even in these really kind of mature pieces of myelin. If you stop MBP expression after a few weeks or months, then I think, yeah, at least a month, I think you do start seeing sheaths degenerate and shrink. So it seems like, yeah, but you do need to kind of continually be producing new myelin, even not in a remyelination context, but just normally. So yeah, it's possible that netrin's just not doing much during development, but it is important for this renewal process. And I guess that would be more the maintenance side. But yeah, it is also possible that it's something to do with plasticity, that maybe, yeah, again, in motor learning and things like that, we know that myelin changes. So if netrin's important for that type of process, then maybe, yeah, the myelin is forming fine initially, but then as it's trying to change and adapt, it's unable to properly reform or stick back to the axon. So yeah, I think it'd be really interesting to look in some kind of plasticity paradigms, either in vitro or in vivo, and see if the netrin the netrin mutants are unable to undergo this. And I think that's kind of what we're seeing looking at different time points in vivo, is that yeah, it gets worse with time, but I'm not sure if we've been able to really nail it down as to whether it's learning or not.
Anyway, thanks. That's a really nice presentation.
Thank you.
Good job.
Jack, do you wanna?
Yeah, so nice presentation. Darian, I almost was going to break away to go do the experiments that you were guiding us from. And I think and in some ways I was going to ask how you're going to bring your data to the public, but maybe I don't want you to. I think we'll take advantage of it.
I'll try to set you up with
The
it.
area that strike and in some ways, from our work that you sort of brought out, is that the notion about whether as you looked at the lineage, that you start with your early progenitors and they go through a development and then they myelinate, whether certainly in the human brain, and I suspect in the rodent brain, you've actually got cells there at different stages of development. And that in fact, whether we didn't do the experiment with netrin, but we've done it with everything else. Tim, we got to go back to the netrin. That we've just done it with fibrinogen, for example, where if we take the earliest cells we can get, which are pediatric progenitors, that they don't like fibrinogen. If we give it to more mature cells, they like it. And whether the same with what you're bringing out is the way you've done it by looking through development, in fact, to bring through the notion of there are different cells at different lineages actually sitting in the brain, and that how one can examine that. And we can do it in the human system. And obviously I'm going to get at Tim to how we do that thing. So I think that's sort of one type of question that I have. If you showed it almost in your second or third slide, where you took a progenitor cell and then you end up with myelination, that if you're going to use those cells, so you need those progenitors, they have to be recruited. Then what happens? When do they start to differentiate? Where's the key there? And if you had a cell that was partially already differentiated, do you think it can take part in the remyelination process? Do you really need those early progenitors to make the things work?
Yeah, that's an interesting question. Yeah, and it's tricky to say. Like, I guess, I mean, in the netrin case, it's complicated because one thing I've never been able to really nail down, yeah, is how much of the heterogeneity is due to, yeah, different cell types versus different stages. And I know in these cultures, for sure, there are different stages. They're not all synchronized, and sometimes the cells are differentiating really quickly, and other times more slowly. So when I'm putting netrin on at later time points, or early versus late, it's not so much that it's affecting all of the cells negatively and then all positively, it's like even early on, some cells seem to respond positively. So I think we really need a better understanding of how oligo heterogeneity is working. I think we need to, I think there are some subtypes with, fibrinogen or netrin or whatever else that maybe are responding early on, and there's others that might be responding later, but it might just be a single subtype that's responding, throughout its whole lineage. I think I need to get a better handle on that, and that's going to require, I think, going back to the transcriptomic stuff and finding out, finding the markers we can use to identify these different subtypes and seeing how they're responding then to these different cues. Really, ideally, I mean, we could stain these kind of cultures for some of the markers that have been described for the different oligo subtypes and see, are the cells that are getting bigger with netrin from a specific group?
So from your reading of the transcriptomic data, literature, what subtypes have we found subtypes of oligodendrocytes?
I mean,
Or do you think that the only real different subtypes are different stages in the lineage?
yeah, I'm become a believer that there's real different subtypes, and a big reason for that is because we see huge heterogeneity in morphology even when they're relatively mature. So even at like 10-day cultures, when they're all kind of seem as big as they're going to get, there's a massive amount of variability in the morphology. Sometimes they grow very, very close, dense pack around themselves. Other times they extend huge distances. And so I struggle to imagine how that's possible unless there's some sort of intrinsic difference. Um, but yeah, we haven't drawn the connections between the different morphological groups or functional groups and the transcriptome. And I think that's the big need right now, is to see which cells are yeah, we need to know what the functional consequence of this heterogeneity is. And yeah, I think we're gonna have to
I don't know that I
look
want to
into
take you on a debate here, but if we change the growth conditions, take away a little bit of the glucose, we can change the morphology and get all sorts of subtypes. So I think so far the lineage is the story.
Yeah, but are they?
Uh,
Yeah, yeah, no, it's
Now,
great.
and then the other favorite question I've asked you before, and I'll say, I mean, again, you showed a wonderful picture of how myelination occurs, showing an oligodendrocyte with one arm, but it's got 40 arms.
Mhm.
Are all the arms doing the same thing at the same time?
Mm.
And
Yeah,
you know, again, you've talked before about the local conditions being somehow each arm seems to be able to respond and what's going on at a very local area. And I find that fascinating, but I don't.
yeah. Yeah, no, I find that super interesting too. So I'm thinking, I mean, there were a few zebrafish papers where they were looking at myelination in zebrafish spinal cords, where you can really clearly see the axon and the oligo. And in these cases, they were looking at calcium activity and as well as like the rate of sheath formation and change and things like that. And so in that system, the cells had only like a 12 to 24-hour period where they were producing like all of their sheaths. And I think people have shown similar things with mouse oligos in vitro. So maybe they're not exactly synchronized, but it is still within the same stage of the oligo. It seems that it's doing all of its sheath growing. I don't think you'll get like one sheath forming and done, and then it will start forming another sheath, you know?
Right.
And yeah, you see these calcium waves that correlate with the sheath growth, and the cells seem to be in modes where they have like, I think it was fast calcium waves initially when they start wrapping, and then the calcium slows down. And when they were manipulating this, they were completely messing up the sheaths. They were either getting like way too small or they weren't forming in the first place. So again, I think the cell is getting into, yeah, it has a myelin wrapping mode, and then it has like a,
Right.
you know, yeah.
Okay, let me ask you one more serious question.
Jack,
Oh, go
before you
ahead.
go on, let me just continue that a bit. So Darian, though, your data has all, like, embedded in your data is the really interesting finding that oligos seem to make a certain amount of myelin,
Yes, yes,
and
yeah.
you have a few really long sheaths or maybe many more short sheaths.
Yeah.
And then there's clear data in vivo that one oligodendrocyte, like even from 100 years ago, one oligodendrocyte can wrap a thick axon and have a really long internode, but
Mhm.
the same cell can at the same time wrap a thin axon and have a really short internode. So you've got the local regulation of myelination on different arms. And and do you think, and some of the 3D reconstructions that have been done over the last few years have made the same argument that oligos seem to be, they seem to make a, you know, if you have a lot of long internodes, you're going to make fewer than if you have a whole bunch of short ones.
Mhm.
As if there's a saturation amount of myelin that can be generated. So do you, if you, like, I find myself thinking about things like homeostatic plasticity, where that synapses, where you've got the ability to modify different synapses, but overall the cell kind of scales itself to a certain point. And
Yes, yeah.
something similar is happening in oligos, or?
Yeah, I think that's a great analogy. I think it's yeah, that is definitely how I think about it. Yeah, they have a finite amount of myelin they could produce, but locally they're deciding where it should be prioritized. Yeah. Yeah.
Yeah.
And so, but at the same, yeah, at the same time, I am really curious about seeing the big increase in MVP expression
Yeah.
centrally, like throughout the whole cell when one process encounters a stripe. I was really not expecting that so much. So I think, one process, maybe one process can encounter a queue and then that gets the whole cell in a different mode kind of thing. But yeah, then there clearly still is a local factor that will determine how much of the resources it's producing goes to that
Yeah.
one point. Yeah.
I'll just say, I hadn't seen the latest version of that data, and it was a good thing I was muted when that came up on the screen. That was pretty exciting.
Nice.
Go ahead, Jack. I just wanted to extend
So
that.
I want to ask you one serious question, then one non-serious question.
Great.
The serious one is that how you're going to put all these observations together that the public can benefit from it. Do you see that someone would have to look up your PhD dissertation and sort of take out what you've written in there? Or do you see that you're going to write some killer review article or still time for one or more original papers? Where is this? I mean, obviously, I'm going to dig in, I'm going to take advantage of it, but how are you going to share this information? That's
Yeah, so yeah, well, I'm doing a manuscript-based thesis. So yeah, I'm written like all the codings results are written up now, and I'm kind of in the middle of writing up the Netrin results. So yeah, for my thesis, they're going to be kind of mock paper, but they'll be paper structured. Afterwards, I'm going to have to do, I think, a little bit more refining for the Netrin results. Mainly, I want to flesh a couple things out a little bit more. But that was, so yeah, both the coding one we're going to try to publish very soon, and the Netrin one probably not going to submit it before the thesis, but I think right after we'll be submitting all of that. And, yeah, so I think, yeah, but both will be published for sure. Yeah, both the projects,
And
I kind of split them up into.
I do sense, coming to the end of your talk, I really did sense the opportunity for one or two review articles. Like there's a lot of interesting stuff there.
yeah.
Yeah. I mean, I know, yeah, maybe
Yeah,
maybe
you
there's more
raised
to be
some
said.
really neat concepts that I'm not sure how they're going to come through in the usual paper or, okay. Now, if you were to take me down to the animal colony, can you show me an autistic marmoset?
Like honestly, kind of, yeah. Like
Yeah?
they exhibit the so a lot of the same like the stereotyped behavior thing, and as well as antisocial behaviors. People have looked at like failure to pick up on vocal cues in marmoset. So yeah, like much better than mice at least. Probably nothing's perfect, but I think they're going to be a lot better than mice.
I don't want to get into the gender issue of marmosets. I'll leave that.
Oh no.
So, Darian, in your atlas and with your immunostaining, can you identify progenitors?
No, this was just more for stains for myelin and axons and different cell compartments and things. We had a few like fluoromyelin and MB. We're staining myelin in a few different ways, but yeah, not progenitors. But yeah, I think
You
now that
could,
yeah.
like it hasn't been done, but you could.
Mm-hmm.
Right.
Yeah.
Okay, I'll stop here.
Great,
Okay.
thanks.
Um, I am okay with questions. I guess I should open up so if anyone in the audience has any questions or any comments for Darian. I'll just reiterate that I thought it was fantastic talk, and it's great to see the data brought together like that. Like really nicely presented.
Great, thank you.
I think at this point, what we need to do is move everybody into a waiting room, and me and Jack and Ed will have a short meeting, and then Darian will call you back in.
That sounds great. I am just trying to find the little control panel for Zoom now. Let's stop sharing. Do I need to move everyone or how does that? I'm not sure.
I think you do. You're the host, I think.
Okay, here it is.
Oh, but if you move them,
Then you
Can I,
won't be able to.
can I
no.
make you the host,
You have to make
Tim?
me
Yeah.
the host.
Yeah, you should make Tim the host.
Yes. Make host. Okay. Okay. It's in your hands.
Okay. So let me see.
The other thing I recommend is maybe we stop recording at this point.
Oh, sorry. Yes. Yeah.